Talking from the consumer's perspective, the quality of meat is related to its chewability, that is tenderness, color, appearance, and very importantly, the microbiological status. As we have already discussed about the basic quality parameters of meat, in this lecture, I shall be telling you about the post-slaughter quality deterioration in meat and their restoration options. Operational hygiene remains the key factor for ascertaining microbiological quality of meat. This includes hygiene in the abattoir environment, the devices used for stunning, dressing, and cutting of the carcasses, the carcass and meat cooling, and finally the packaging area. Also, the packaging materials need to be clean and hazard-free. In meat industry, major part of the contamination occurs from the use of non-sterilized dressing and cutting devices, especially the knives. Regular disinfection, sterilization of these knives and the other devices is therefore very necessary. It is a general practice to store the dressed carcasses in cold or refrigerated environment. Lowering the temperature inhibits microbial growth. Also, during this stage, the proteolytic enzymes, primarily CALPAINS, cause degradation of the muscle fibers, imparting tenderness or softness to the stored meat. However, this cooling step also requires some careful pre-considerations. Cooling errors like cold shortening and thaw rigor must be avoided by such precautions, and the cooling efficiency can be enhanced by treatments like hot or accelerated processing. All these will be discussed in the next slides. The first is cold shortening. Shortening, as you know, in case of meat, is referred to contraction of muscles or meat. As I have told you about the importance of ions in the regulation of muscle contraction and relaxation, in living muscle cells, calcium ions play important role in muscle movements. The excess of calcium ions is trapped by the sarcoplasmic reticulum present inside the fibrillar muscle cells. ATP produced by the mitochondria assists in the muscle contraction. So calcium and ATP are the two factors responsible for contraction of muscles. After that, the ATP level in the cells gradually decline, and after some times it disappears completely. It gets completely used up. Also, when stored in cold temperature, the sarcoplasmic reticulum cannot entrap calcium ions and anymore. They cannot entrap calcium ion anymore, and the ions are released in a high quantity in the cell cytoplasm or sarcoplasm, as in case of muscle cells. But if the dead animal's carcass is chilled or cooled soon enough, when it is still synthesizing ATP, that is, the rigor mortis has not started yet, and the pH is still about 6.2. You know the pH it goes down uh, from near neutral to about 4.5 like that. Sorry, 5.4 like that. So before the rigor mortis has occurred, if you are chilling or you are cooling down the carcass, then major contraction occurs in the cells. Available ATP and high abundance of calcium ions in the sarcoplasm are responsible for this. This contraction occurring due to immediate chilling of fresh carcass is called cold shortening. It is more prevalent in red muscles where mitochondria per unit cell is high in number, producing high amount of ATP. In white muscles, production of ATP is naturally low and problem of cold shortening is almost absent. The problem of cold shortening can be avoided by certain precautions. The first is by delayed chilling, that is, letting the carcass use up all the available ATP and letting the pH go down to below limiting level of 6.2. The chilling temperature may be kept higher, that is about 10 degrees Celsius. The cooling rate be kept low, that is slow cooling down. Once cold shortening has started, electrical stimulation can be used, and more popularly, stretching, which is generally hanging of the carcass with a hanging hook, is used in diabetes to soften the tightened muscle caused by the cold shortening. This significantly stretches the muscles. Hanging of the carcass upside down, as shown in the slide, it significantly stretches the muscles, not letting them stiffen or harden during chilling, so keeping the tenderness intact. The next is thaw rigor. It occurs under similar conditions as cold shortening. Actually, thaw rigor is a result of cold shortening. It occurs uh, by cooling of the meat carcass before the start of rigor mortis, as in case of cold shortening. As discussed in the last slides, cold shortened meat are those which have been chilled before rigor mortis had started. Now, when the meat is brought to room temperature again, rigor mortis tends to occur. The rigor mortis which was not allowed to occur initially, and the material was stored and the carcass was stored in chill environment, after bringing it out to the normal temperature, rigor mortis starts to occur, causing further changes in the muscle structure, and change in muscle structure results in change in the muscle texture, and this often results in significant drip loss. Drip loss is the loss of liquid or the watery substances from the meat. This characteristic is called thaw rigor. Prevention of cold shortening also prevents thaw rigor problems. So, the techniques applied to inhibit cold shortening are the techniques to avoid thaw rigor also. Cooling has to be efficient and uniform as it accounts for significant power cost. So, in big abattoirs, the refrigeration system it involves high cost. Presence of bones and fats, which are actually wastes or byproducts, often make the cooling of carcass non-uniform and costly. In hot or accelerated processing, the carcass is deboned and defatted before subjected to cooling. This has a number of advantages, as a more uniform and suitably rapid rate of cooling can be attained after removal of the insulating bones and fats. All these parts are removed. The cooling space per unit mass will be lesser, so the requirement of space for storing after deboning will be lesser, obviously. Hot deboned meat, the deboning is done at room temperature. The hot deboned meat has a better water hydration capacity. That is, it can absorb and retain more water than normally cooled meat with bones. And due to uniform cooling, the chunks of meat attained after this hot or accelerated processing, they attain more uniform color. So the color is also better in case of hot or accelerated processed meat. However, due to removal of the bones, parts of the meat cuts, especially the joints, lose their characteristic shapes. A sort of toughness develops after cooling. So these hot processed meats are a bit harder. Also, as the deboning is carried out at room temperature or above room temperature, the risk of microbial contamination and microbial infestation is higher, and process hygiene is of utmost importance for the deboning process. Apart from the discussed pre-slaughter and post-slaughter treatments to prevent toughening of meat, there are a few other techniques that are used to soften the meats. The first one is stretching by mechanical devices which do not let the chilled meats or muscles to contract. The disruption of myofibrils by enhancing the activity of endogenous proteolytic enzymes or endogenous protein degrading enzymes like CALPAINS and CATHEPSINS. These enzymes activity can be enhanced by either a long time aging at low temperature or a shorter time aging at high temperature. Electrical stimulation can be used to rupture the lysosomes of cells which contain cathepsins. Electric shock also causes violent contraction of muscles, tearing down the myofibrils and loosening the meat. Calpain enzyme activity can be also enhanced by injecting calcium chloride solution in the carcass. So if you are injecting calcium chloride, the activity of the calpain, the endogenous calpain enzyme will be higher. And apart from the endogenous enzymes, addition of plant-derived exogenous enzymes like papain from papaya, bromelain from pineapple, ficin from fig, and actinidin from kiwi can also be additionally used to degenerate myofibers and thereby tenderize the meat or soften the meat. So with this, I conclude this lecture. Thank you very much.